Oh, can someone get the door?
Yeah, again, my sticker. So now we're off. They got up in there all last year.
Everyone have their cell phones turned off? Or vibrate. Okay, so I guess we'll get started this afternoon. So welcome to the second season of the Saskatchewan Prairie Conservation Action Plan Native Prairie Speaker Series. My name is Michelle Clark and I'm the PCAP manager. I'd like to thank the Government of Canada's Habitat Stewardship Program for Species at Risk for their financial support, and the Saskatchewan Watershed Authority and the University of Saskatchewan College of Agriculture and Bioresources for their in-kind support of the series. Our speaker this afternoon is Ian Phillips with the Saskatchewan Watershed Authority. Ian is an aquatic invertebrate ecologist with the Saskatchewan Watershed Authority working on management of aquatic ecosystem health in his position since 2006. He has a Master's of Science from the University of Alberta and a Bachelor's of Science from the University of Regina. Currently, Ian is enrolled in doctoral studies at the University of Saskatchewan in conjunction with his position with the Saskatchewan Watershed Authority, focusing his dissertation on the interaction between the environment, human activity, and aquatic community ecology in the Northern Great Plains. His presentation today will be on insights into the relationships between crop agriculture and aquatic ecosystem health in Saskatchewan's Northern Great Plains. Would you please join me in welcoming Ian to the stage?
Thanks, Michelle. This might give a bit more kind of expectation that there's a large relationship between crop agriculture and aquatic ecosystem health that I'll come to describe. And a lot of what I'm going to pose to everybody is kind of a concept of an environment that's already quite disturbed. So I'll get right into that. And the take-home message, the Northern Great Plains, based on our research, are turning out to be, and as a theme, quite an extreme environment for aquatic organisms. I'll go through kind of a description of that with some description of both the environment and the bugs that typify it, and how expectations for tools to measure that ecosystem health are different here than they would be in other ecosystems around North America and the world where typical tools for aquatic ecosystem health have been developed. And finally, just propose a concept for discussion or maybe some feedback that we need to adjust because of this, what humans want and what's natural and what's pristine, and have a bigger discussion on that. Our expectations seem to be that European agriculture has been a negative thing, and in some cases I think that can be well argued. But in these aquatic ecosystems, put forward that it's a disturbed habitat to begin with, and our value systems that we place on it might be best suited to what values that we want, the goods and services that we want to recover from, such as increased biodiversity and characteristics. So I'll get right into it. So just to begin by typifying how it's an extreme environment, this is the Frankston River down in the south in the province, and on a day where it's hotter than the hubs of hell, and in this stream it slows right down midsummer, and the organisms that live in there have very little relief from the riparian area. This is there's very little shading, not a lot of contribution of lox and detrital material to shade within the river, high production of macrophytes, and it reaches quite high temperatures in the summer. This is a figure showing the daily maximum temperature over the course of a few months in the summer of 2009. I trimmed down the data and focused on maximum temperature just to provide a clearer example of how temperature differs between what would be a classic pristine stream and on the prairies what's a pristine stream. Here, the prairie stream is characteristically about 5 degrees warmer than a stream that is in the Cypress Hills or boreal type stream that has some riparian infrastructure surrounding that stream. So it's warmer in the summer, and in the winter, these organisms, whether in the boreal or the prairies, but on the prairies, they have to suffer a lot of scouring, low temperatures under the ice, commonly low oxygen. It's quite a harsh extreme during the summer, and on the flip side, quite a harsh extreme in the winter. It hard for us working as well. Hopefully, would be here. everything thaws, it doesn't get terribly much better. And often in these ephemeral habitats, there's not a great amount of runoff, and it only happens for a short period of time during a freshet. You end up with water bodies that at best are wet, more of a damp crotch in the landscape than anything else, and it's pretty stagnant. There's not a lot of oxygen there as it is, and the only reason there is oxygen is really because it's cool. Once it starts to warm up and production rises in that more of a lentic habitat, oxygen decreases, and a lot of the characteristically positive organisms for ecosystem health won't be able to occur in our reference sites as it is because of those conditions. At best, it dries mid-summer and starts to vegetate over and becomes a terrestrial environment. And the organisms that live there then have to deal with both extremes in the summer, extremes in the winter, periodic drying, not only consistently on an annual basis if you adapt to it, but over longer climate series, you have to be able to colonize, have resting stages. The organisms need to be highly tolerant. This is from John Arthur's watersheds area down in the southeast, where it essentially turns into a terrestrial environment once the freshet has gone through and trees over. If we go up a scale from these ephemeral streams, ones that do maintain their water, a couple stream orders up, they typically slow right down and become stagnant and resemble more lentic habitats than lotic habitats, and challenges that bugs exist with are different than those if they're in a more classic mountain stream or a boreal stream. This is probably the best example of a reference site that's flowing through the summer, and it doesn't have a lot of riparian vegetation. It's subject to a lot of solar radiation and all the other vagaries that come along with it. What we're trying to do with this work, and the reason I have my job, is essentially to identify, based on all that background, how does agriculture play an influence, if at all, on the aquatic ecosystem health? And what should our expectations be to set objectives for what we want ecosystem health to do up in those streams? And this is where I propose to everyone how consider the concept of what exactly is pristine. Prior to European settlement, this was quite a disturbed environment as it was. There was frequent grass fires from our Aboriginal, naturally, and from our Aboriginal peoples who lived here. And there was also high amounts of bison activity, which would have, I'm not saying identical, but comparable effects to having cattle activity on the landscape. It's all focused around a scarce resource like water. It would be a highly, an environment subject to a lot of disturbance to begin with. So now we have to take all that knowledge and build tools for assessing this aquatic ecosystem health. And what I'm showing here certainly is not a very extreme. This is typically where these tools were developed, in high relief areas with cobble riffles. And if you're looking at a pristine site or one without human influence, there's sites that have a lot of organisms that are very sensitive to human change. So any increase in the amount of organic pollution, pesticides, things like that, and those bugs suffer that are sensitive to that. The bugs start to become more prominent in their abundance, and we're able to see distinct community change and delineate those changes in a pretty descriptive way. This is a colleague of mine, Michelle Bowman, in the foothills of the Rocky Mountains, in a typical stream where these sorts of tools are developed, and using a method that was developed for those streams. This is the type of breach you use, where it's a riffle habitat, and it works quite well for a reference stream. However, on the prairies, our riffles are wildly different, really. If we have them at all, they desiccate through the year. As well, they're a low point that's stable, and they're subject to both cattle crossing and human activity, and that kinetic energy passing through there disturbs the habitat even more relative to what the rest of the condition might need in that stream reach. And assessing this habitat might be a red herring for what the condition is in that river because it's focused activity. That, and well, a lot of these regions dry right up once it comes to the period of the year when you typically sample them in the fall. In the mountains and in the American Northeast, those streams would run more consistently into the fall, and here that might be an inappropriate time period to be sampling. sorts of critters there looking for bugs as well. This is the best you could probably hope for, where it's wetted with, and as a reference site, has a lot of intact vegetation. And you can use a run to assess this, and I'll go into the tools that we use currently. However, reminding everybody that in conversation and looking at historical records, riparian vegetation may not have been as intact as it is today. So this reference site might in fact be something different than prior to agriculture and original settlement. And this is a critter who typifies characteristically positive ecosystem health in the classic approach that was developed for bioassessment. This is a caddisfly, Limnicoidae is the family. I'm not quite sure the genus offhand, but I've chosen this picture because it personifies what's occurring on the prairies where they're adapted to a lot of human activity. This solitude absolutely flagging taken for this case. Furthermore, we have top organisms in these aquatic food webs for the Central American Vertebrate Assembly that are not included in other monitoring programs around the country because there seems to be less important, less dominant, don't occur as frequently, and a few other methodological reasons. But in Saskatchewan, crayfish, for example, here forms a dominant part in these food webs. Further, we have a lot of organisms such as this Lamina stagnalis and this water scorpion who are adapted to low flowing environments and would be excluded in the other tools that are used around North America because they reflect limbic habitats, not necessarily flowing within streams. We do have a lot of diversity though, and it being said that we don't have the same sensitive species that occur in the American Northeast or in the Rocky Mountains, we do have community change with activity. Even though agriculture comes along where we have bison come through, it's not like bugs disappear. The bugs change, and we have species that are adapted to these harsh conditions. The challenge that we face as the watershed authority or surface water managers is figuring out how they change, what that reflects, what goods and services these organisms provide that have occurred and arrived since that change, and whether or not it's desirable. This is a Asian emerging down in the Souris River near Rafferty Dam. And here's another eating a smaller dragonfly. Further, these chironomids, for example, these are midges. You'll come across these in the summer riding your bicycle through a park where your eyes and nose get full of tiny bugs. They're typically used as a coarse example and a coarse metric of a lot of human activity. As a family, the chironomids express a lot of disturbance and are very tolerant of a lot of disturbance. So if you find lots of them, say, in the Rocky Mountains, you'd be able to make conclusions that this is a highly disturbed environment. In Saskatchewan, they form a dominant part of the abundance in our assemblages, even in reference sites. However, that being said, some of the tools we adapt is looking in that family at the different genera that occur, and we find that there's a lot of diversity, and that they themselves, as a taxa, respond to these changes, where in other parts of the world, we focus exclusively on the coarse taxonomy of chironomid day. So they're quite useful here and strong record for the future. So too with things like black flies that occur in high abundance in some of our habitats. That's just a picture of a horse I thought I'd get over. So the reason I have my job is to provide, now Heather, it's a measure of change here.
That was
And within the State of the Watershed report, this is covered from the 2007 report, and SWA recently produced the 2010 report, right, Heather? Yep. For 2010. And it's to express ecosystem health and provide a measure of the health of the water bodies for the people of Saskatchewan. It goes in concert with riparian health or water quality to provide a picture of the surface water resources we have. And now I'm going to get into some of the heavier stuff of this work. I've given you a description of the types of constraints we're working with, some of the unique characteristics. Now I'm going to go into how we're dealing with that and some of the methods we've developed. Essentially, what this all boils down to is walking into the stream with a net and collecting bugs. And it's pretty straightforward. It's not rocket science. Here characteristic of the type of morphometric or morphological components of the organisms that we'll look for in order to figure out who they are. Once we've collected those bugs, we use remote sensing to evaluate the land use in these immediate areas. And what I'm presenting here is essentially the first cut of our bioassessment tool we built, essentially the feasibility study. Here you'll see this is the plains around Regina, and each of these light blue dots is a site, and the purple area is the upstream catchment. It's a buffered area that we evaluate the land use of at each of these sites. What we found quite early on in assessing stratified random positions throughout Saskatchewan's rivers south of the boreal is that there's a high amount of human activity. There's very few areas where there's low amounts of crop, low amounts of cattle, essentially where we could use the land for plowing, putting a crop in, or putting cattle on, we did. And the criteria we came up with to find the best possible sites to represent the least amount of human activity were quite liberal. Less than 50% cropland, for example, less than 50% pasture land, and a total human land use down here in the bottom right of less than 80% total human activity. That's a summation of all the different activities that occur in that came from the Aquatic site. This data that I'll present is essentially a feasibility study of 200 sites from 2006 and 2007. And it's only the benthic macro invertebrates for a biotic component. This tool can be adapted in the future for algae such as diatoms or fish, that sort of thing. Right now, as the authority, we're focusing on the benthic macro invertebrates, one of the most diverse, one of the most common, easy to identify. At each site, then we evaluate the land use and the underlying physical characteristics, water chemistry, and habitat. And it's important to focus on the underlying physical characteristics. Keep that in mind when we go as a point in the site. We include the stressors evaluated to the land use as the reference site is selected to be the best possible condition to find. And then this is where it becomes important for the underlying physical characteristics. We take those reference sites and we evaluate the assemblage community similarity, or assemblage similarity. Here on the y-axis we have similarity, Gray curve measure of it, and on the x this is our sites. And we group them by most similar sites to each other. So essentially it breaks down into a dendrogram. We can get this pocket of sites is most similar to each other for the assemblage that it has relative to this pocket and this pocket. We let the bugs explain what defines who they are and where they occur in the landscape. So once we've done this, we figure out, okay, why are these organisms more similar than these ones? What are the underlying physical characteristics that define who occurs where? If we were to take reference sites from the boreal forest, for example, and compare volcanic tree tongue with a plain, the underlying physical characteristics of those two sites are wildly different. And we would never logically expect that the bugs that occur on the giant plain should look like the bugs that occur in the boreal forest, the two different environments. But we have to identify what are those underlying characteristics that define who occurs where to begin with. Once we do that, then when we have a test site that's impacted, we look at those underlying physical characteristics and we're able to match it to the reference sites that it should look like based on those underlying characteristics it has. A good example of why this is important here is just an ordination plot of assemblage composition correspondence analysis. We have these little X's in the difficult to see, but they're test sites essentially. And then we have the reference sites. Here in red is group 2, here in green group 3, and in blue would be group 1 from our reference site similarity analysis. If we were to compare this test site to group 1, it would come out as impaired if it has, if we were to compare to those sites. But if it was compared to group 3, it comes out as being within reference. It's quite similar to those organisms in group 3. So they have to make sure you match apples and apples and oranges so your expectations are set. What we found is that geology, the underlying superficial geology of the site, and ecoregion were the underlying physical characteristics that best explain the bugs that are present in our reference sites, and would ergo define what we expect them to be in our test site. And here's just a kind of a map, a graphical example of where these sites occur. In yellow here, that's biological grouping 2, and in green biological grouping 1, and red biological grouping 3. I apologize for pedestrian names I use, but I created defining biological ecoregions based on the bugs we have. But for all intents and purposes, you can see biological grouping 3 is more of a boreal transition or aspen type of community. 2 is more aspen mixed, moist mixed grassland, and grouping 1 has a lot of geologic characteristics that define it. We'll look at exactly now. Now, why this is important in evaluating the metrics is that in some of these groups, these benthic macroinvertebrates respond differently. If we were to have, for example, here diversity on the y-axis, this is a measure of the number of species of organisms at a site relative to their abundance. In biological grouping 1, there's a very little interaction between this diversity measure and cropland. You can increase cropland by 1, and diversity stays the same essentially in these sites. So as a metric, that's not a very sensitive metric to use for looking at the impact of agriculture on ecosystem health. However, in the boreal transition here, biological grouping 3, there is a relationship, albeit weak, but there is a relationship between diversity and cropland. So if you increase the amount of cropland, you decrease the amount of diversity. Just an example. Now, to take a kind of a case example to show everybody how this has been applied in order to make conclusions about ecosystem health at sites, I chose a site on the Copeau River in the northeast, using diversity as a metric for this change because of agriculture. So there's a test site that we selected in 2007, and we determined that it's significantly different from reference site. So how we determined that, we'll forewent a lot of description of the statistical methods, but essentially we look at whether or not the site falls within a 95% confidence interval of the reference site given in space here. You can see this test site in this hypothetical example is quite lower in diversity and, well, mid-range for abundance, but lower in diversity than the grouping of reference sites. It falls outside of the 95% confidence interval to 99% confidence interval. It's quite significantly different for diversity than the reference sites that they're compared to. This is just to provide the logical flow of progression. You go from collecting the bugs, you evaluate the distance it is from your reference sites and diversity, and you evaluate whether that difference is significant. If it is significant, you can make conclusions that it's significantly, ecosystem health is significantly impacted. In this case, it is significantly impacted, and therefore it is impaired. And you can scale back from this test site analysis, it's called, to identify which of the metrics, which of the metrics is causing that impairment. Here's a summary where the condition is overall impaired in test site analysis, and we can look at the different components of that impairment and say that, abundance is in green here to represent it's not impaired relative to reference sites. Richness is okay. Community composition, that would be correspondence analysis, but the community is not that greatly different. However, diversity is greatly decreased at this site. We then look at our previous slide I showed there. What does diversity mean? Well, it has a negative correlation with percent agriculture in the sites, and we can diagnostically take it back to try and identify which stresses on landscape are causing that degradation. And there's multiple questions afterward. I thought I'd just try and keep it moving through this so we can get to some discussion about it. The recommendation we make from this then is promote management and mitigate that crop-related stress at this site. That might bring this, set an objective, bring that diversity up to a diversity that reflects the reference sites in this biological introduce everybody to the second component we're pursuing with nitrogen. I'm bringing this in to try and express and reinforce that we need new tools in order to assess ecosystem health in the prairies. This is one we're developing in concert with the University of Regina. And it has to do with the stable isotopic composition of organisms in the water body itself. Essentially, you are what you eat with nitrogen and carbon. You reflect what's in your environment. And as you go up trophic levels, you increase the ratio of the two nitrogen isotopes, N14 and 15. You change that ratio in your tissue. Using this concept, if there's more nitrogen in the environment, we have a bit of it can cause problems. Just to provide a case example with Wascana Creek down in Regina, there's a lot of nitrogen coming in from the wastewater treatment plant, and it comes in the form of ammonia in the winter, and it causes some significant impact on the biological condition of that river reach. We're trying to adapt this to that problem. We'll hopefully be able to use this as a tool to diagnostically point at which, in this case, nitrogen as a stressor that we need to mitigate in order to improve ecological condition. And how that decision making essentially, before it works, is you evaluate in the beginning with a pesticide analysis, is biotic integrity impacted, yes or no, and if it is, then maybe management action is necessary in order to improve that ecosystem health and return to something that is desirable. Now you evaluate the M15 to see reference levels. Is it higher than what we would expect at reference levels? And in the case of where we decided that biological integrity is impacted, if it does, then maybe we need to target nitrogen sources. If it doesn't, then maybe we need to look at other metrics. There might not be nitrogen, it might be habitat degradation, pesticides, other problems in there. If it doesn't, if it's healthy, essentially, we don't need management, it probably doesn't matter whether or not nitrogen is seen because that site is able to handle it. But some of the methods behind this are using primary consumers such as mussels and snails, which reflect a lot of the particular organic matter and algal production at site 9 and 15 in those sources, and we process their tissue to the N15 concentration. Here's a conceptual presentation of how we pursue these results. Essentially, we take our site, for example here, Wascana Creek, Wascana Trails, and this B is a reflection of the pesticide analysis, and it's high, 17.6, and it's significantly impaired relative to reference site. Diversity is okay, at 1 there, it's not significantly different than reference. However, abundance is less than 0.05. It's significantly impacted. So too is the stable nitrogen isotope level in the tissue. Back to that decision-making flow chart, nitrogen is potentially a source of impact in that ecosystem, a source of human activity that could be mitigated in order to improve ecosystem health. However, percent EPT, that's ephemeral triple phosphate, is a classic description of African vertebrate health, isn't impacted. This is a good example where on the prairies, this isn't very sensitive at all to human activity. The type of organisms we had, if we go back to the beginning of the talk, aren't very sensitive to organic pollution. They're already in a high amount of disturbance in their environment. Well, move right to the end here, just to express that in the future, we have expanded this work last year to an additional 102 sites and a couple more this current year. And we'll have more resolute results, a finer precision for reference condition, and we're pursuing this in the development of better tools through my graduate study at the University of Saskatchewan. And we'll be focusing on at what point does condition on the site depend on the watershed above it, whether you can focus on the immediate area 2 kilometers upstream, 10 kilometers upstream, and moving right to the very end. This is one of the funnest jobs I can imagine anybody have, and certainly anybody's Prairie Adaptation Research Collaborative, Sask Environments, Fish and Wildlife Development Fund, that sort of thing. Kevin Kirkham and Deanne Schultz were instrumental in putting this together. Really worked for 4 years, and since then I've had a variety of other helpers who have done a great job with this. Anybody have any questions about that? I know I kind of sped up towards the end, Michelle, giving me a signal that I've done time.
You were talking about how you were collecting your samples by using CNET.
Yep.
And I'm wondering how you managed to make that an accurate way to measure all of this.
Great question. So in any of these programs, whether it's the national program, Cabin, Environment Canada, or the American programs, they focus on standardizing the type of sweep you do, the amount of area, amount of time to sweep at each site. So it doesn't necessarily reflect density of bugs at that site. What you're trying to do is keep what you do at a site consistent between sites, so you can compare relative abundance and relative diversity. And what we did at our sites was 5 spots in the transect along perpendicular to the direction of flow, and accumulated that all into one sample, and then did that consistently across sites, so that when we give a measure of abundance, hopefully how we measured abundance at site A is from closely identical to how we measured it at site B. It differs a bit from what Cabin does, which is in the ripples, and they use a time sweep, because we had to adapt to the conditions we find on the stream. If we were asked to evaluate, say, the ecosystem health of a prairie province waterboard site on the border, we're asked to look at that location. We can't really go, we can't do it because of the ripple, and can't do it because when we are trying to adapt through other tools to our situation. We had to find a tool that worked for the predominant characteristic of the water. So we use a run, and we use those spot sweeps in transect. And we're working with Environment Canada's cabin group to find how similar the communities are between what you collect with my method or SWA's method and what you collect with the cabin method, and see if we can compare the results. We'll have come to this conclusion. However, it's appropriate to still moving rather than. Okay.
And so your reference sites in terms of crop level, you correct that they were under 50% crop?
Yep.
And did you have many at all that were down very low?
There were a common, go after the question from here, a common proposal pitched to me is we should look at Grasslands National Park, for example, and some of these parks that are protected. They're great, only that they capture only a few reference sites, and those streams don't necessarily capture the main underlying physical characteristics in southern Saskatchewan. So we had a few, like at Grasslands National Park, that were, for all intents and purposes, removed from the inventory, and there was some at that other end. What we did in the past, at the very end here, what we did last year, was once we understood, okay, the best we got the first time was 50% or 50% crop agriculture, we prioritized at our sites in the landscape, targeting those that would have less, as minimal as possible. So we set a target of 15% or lower, and provided a giant set of sites and randomly selected from that subset to go out and assess. Site strategies and put that site selected in place. And what we've approved then is a set of reference sites that are much more conservative than what we pursued in the first site.
So I'm wondering, given that there isn't a pristine reference site out there, are you familiar with anything that the is doing as backcasting on what pristine should be, and that might be a useful approach?
It is a approach, it does go into the South Saskatchewan River, doesn't it? Yeah. Please, if anybody has any questions about this book or any interest to provide comment, I'd love to hear from them in the future. And we get it when we do the government directory or Kathy Trinshall has the contact information. Thanks, Lily.
Thank you. Yes, so as Ian was mentioning, his presentation, we've been broadcasting it live today, and it will be available on the PCAP website for future reference. I'll also include his contact information with his permission so that he can be contacted in the future if you have any questions or would like to work with him. Just a quick reminder that we have another presentation tonight at the Western Development Museum. Luke Delaney with the Iwasan Valley Authority will be talking about biodiversity enhancement approaches, the difference between high-quality plant communities versus those under stress in the Iwasan Valley, and that's at 7 o'clock tonight. The speaker series again is free, and so is parking, and the doors will open at 6:30. So I hope to see everyone there. And also, if you visit the PCAP website at www.pcat-sk.org, you'll see a list of the future speakers and topics that we'll have. The next one in November is in Swift Current, and again, we hope to broadcast it live as well, so that you can watch the presentation from your desk. So thank you all for coming, and have a great day.